So tonight we're going to have a topic. It's going to be tech trends from trial to market. And our first speaker is Mike Mulligan, and that will be followed by Heather Evans and then Dave Linker. So first, Mike Mulligan is a professor of cardiothoracic surgery here at the University of Washington. And he is the director of the lung transplant program and also advanced lung disease surgery program and minimally invasive thoracic surgery program. He performs pulmonary endarterectomies. Do you guys know what that is? Okay, so when your blood vessels in your heart get plugged up with blood clots, he cleans them out. It's the rotor-rooter man. All right. Then he's also does lung transplantation and lung volume reduction surgery. Do you guys know what that is? One of you does. Anybody else? Okay, so sometimes when you have certain types of lung disease, like COPD or something, we take out part of your lung and then you can breathe better. It's true. I know, a lot of people think I'm making that up. Is that true? It's true. Okay, so there you have it. How cool is that? Who would have guessed that would be right? So here he is water skiing, which is like way cool. Considered a national expert on video-assisted thoracic surgery lobectomy. A lobectomy is when we take out part of your lung. And leads the regional and national courses on the subject. He also has considerable experience in the management of esophageal cancer because your esophagus is right next to the lungs, so you could see where that would be good. Benign and malignant thoracic diseases and end-stage lung disease. His areas of scientific investigation include studying the mechanisms that could cause acute lung injury following transplantation and regulation of tolerance and long-term lung graft acceptance. Lung graft is when someone gives you their lung. Okay. And when he actually does his surgery, he stands on that blue ball to show that he has a lot of core strength and to make the residents look bad. All right, he's won local and national teaching awards and runs an NIH-funded laboratory, including the Mulligan Award, which was given to a cardiothoracic surgeon who exemplifies outstanding teaching and mentorship in the US. He earned his medical degree at UConn School of Medicine in Farmington and completed his residency of thoracic surgery at Columbia. And then he tootled on over to the University of Michigan, go Blue, for pathology and fellowship in thoracic surgery. I didn't know you were a pathologist.
I dabble.
You dabble. All right, our speaker after that, because that's pretty cool. Our speaker after that is another amazing person. This is Molly Lindquist. And she's bald in that picture, and there was a reason for that, because I imagine that you just had some chemotherapy. So usually we give chemotherapy when you have cancer, so that's my next leap of, deductive reasoning. She is the founder and CEO of Consano, and Consano is a crowdfunding website, and it brings the world of medical research to a platform on the web that enables anybody, to donate any amount, small or large, directly to a medical research project that matters to you. And she actually set up one of these programs for Dr. Mulligan, so amazing. She's a breast cancer survivor, and so when you actually see her get up and speak, she's going to have hair, and her hair came in a really great color compared to my hair. And she wants to further cancer research, especially for her girls, who obviously are very beautiful. And that is why the idea of Consano was born. So here we are back at the UW with our incredible speakers. And so our first speaker tonight is going to be Mike Mulligan. Please welcome him.
As you heard, I'm Michael Mulligan. I'm a professor of surgery, and I run the thoracic surgery unit, which means chest surgery and the lung transplant program. In addition to that, I do a fair bit of basic science research. So what I've been asked to do tonight is to share a project that is typical of the kind of work that's done by clinician scientists, meaning those people that care for patients and do basic science research, to give you a sense of how important and relevant it is, but to also give you some insight into how urgently we need funding, that some of these ideas have finally matured, but the funding climate is desperately bad right now, and we're really on a shoestring budget trying to get these ideas across the finish line so they can start to help patients. I have a philosophy of research, and I have three basic tenets. One is that it's best done when you align your clinical and research activities. In other words, if you're rounding and you see a problem, you're the best person to ask how that problem occurs and what we can do to fix it. So align your clinical practice with the questions you're trying to answer in a research laboratory. Make sure it's relevant. There's a lot of research that's sort of molecules for molecules' sake, and that's fine, that's fundamental, that's foundational work. But we need to be helping patients. Anything we do with medical research dollars needs to help medicine. And finally, embrace unanticipated findings. I think often the reason that medical knowledge seems finite or limited or frustrated is because we're looking at things in the wrong light. We're looking at them from the wrong perspective. So if something sneaks up on you and is directly opposed to what you thought might happen, think about it. Particularly if you can validate that, that may lead to significant discovery and advancement of the science. In lung transplantation, and this is the subject of my research, there are three basic problems. There aren't enough lungs for everyone, so people continue to die on wait lists across the country. Sometimes we put lungs in and we do a beautiful job surgically, we do a beautiful job preserving the organs, and they still don't work. They develop something called acute graft dysfunction or reperfusion injury. Reperfusion means you stopped blood flow in the donor, took it out, and then when you restored blood flow in the recipient, you triggered an inflammatory response. No one sometimes sees it coming. It's not rejection, it's not infection, it's inflammation. And it's a fairly vigorous brand of inflammation. And the final major problem is chronic rejection, like vanishing bile duct syndrome and liver transplantation, or coronary disease and heart transplants. We have something called obliterative bronchiolitis, or OB, that develops in long-term lung transplant survivors. And unfortunately, this remains the leading cause of death in long-term transplant survivors. We don't know how to stop it yet. Sometimes we can stabilize it, and we definitely don't know how to reverse it. This is the problem we're going to talk about tonight, and it's obviously bad. This is a chest x-ray. Ordinarily, your lungs should look like a dark gray color with some fine vascular patterns, but it's whited out, and it's whited out because these lungs are stiff, they're edematous, and they don't pick up oxygen very well. And this is what happens when transplants go bad acutely. Nothing was wrong technically with the way this operation was done, but this patient was stuck on the ventilator for a very long time as a result of this injury. We have a grading system for it, and what we're really talking about is the most severe form, so-called grade 3 injury. And it's also known as primary graft dysfunction, or as I said, reperfusion injury. It is associated with a 30% mortality rate, meaning of the patients that develop this problem, 1/3 will never make it out of the hospital. It increases antigen expression, meaning that there are donor-specific proteins that label this as somebody else's lung, and when the lung gets injured on the way in, you express more of these proteins, and you announce to the recipient immune system that this lung shouldn't be there. Not surprisingly, down the line, there's a higher rate of rejection. And this increases the risk of all sorts of surgical problems like airway healing problems and long-term rejections such as that associated with obliterative bronchiolitis, or OB. Well, most strategies that are currently exist are targeted at preventing it. And this is meaningful. You can't prevent lung injury that develops after a car accident because you don't know when the car accident is going to occur. But you can determine when blood flow is going to stop in a donor and when it's going to resume in a recipient. So you can predict when the insult is going to occur and pre-treat it to prevent it. And preventative strategies make the most sense in this particular injury because it's very hard to stop once the reaction starts. The lung is a vigorous inflammatory responder. I've spent the last 20 years categorizing different mechanisms of acute lung injury, and this is a bad one. Once it occurs, we see patterns of cytokine release. These are proteins and chemicals that are released from cells that attract other inflammatory cells in and spool them up in terms of their injurious activities. They excite the surfaces of lining vascular spaces or blood vessels and airway channels so that white blood cells and other inflammatory cells can stick to them and get into the tissue. And once there, a particular brand of white blood cell called the neutrophil releases all sorts of toxins and causes tissue injury. We excite the vascular space in terms of upregulation of the endothelium and the airway space with the epithelium being excited as well. And even immune cells migrate in and probably form the link between acute injury and subsequent rejection. We've spent, as I said, the better part of a couple of decades targeting different proteins, different adhesion molecules, different cells. And the problem is there's so much redundancy in this reaction once it starts that you only get partial protection. It really doesn't work to intervene after the injury has started either. You need to pre-treat, and selective blockade doesn't work. So we thought, well, if we consider this as an inflammatory pyramid where it gets broader as the reaction proceeds, let's march farther upstream. So we looked farther upstream and we found that the macrophage, one big giant white cell that sits in the airway space, was releasing chemicals that were flogging other cells to respond. This was the quarterback. The alveolar macrophage, or AM, is the quarterback of this inflammatory response. We then found out that there are proteins and genes that are upregulated within the macrophage that drive this reaction. We thought we were getting closer and closer to the pointy end of the pyramid. We actually found that there was something called a mitogen-activated protein kinase, which was an enzyme that would trip on and start the reaction. But we had no idea where the switch was. We were left with a fundamental question: is how is this oxidative stress or this reperfusion stress initiated? Well, when you talk about the way cells respond to stress, the kickoff, the switch, if you will, is usually a receptor. So we postulated that it had to be a receptor, and since the macrophage was the quarterback, that the receptor had to be on the macrophage. And there are certain kinds of receptors called toll-like receptors that exist in humans, even exist in insects. They're very well conserved across species, and they're present all the time. You don't have to be sensitized to have these. We all have them, and they allow us to fight off bacteria and do a number of innate immune functions. There's a specific receptor called toll-like receptor 4 that's been implicated in reperfusion injury of other organs and other types of lung injury, and it is densely present on the surface of macrophages. Not only that, we know what it responds to, and we thought, this is great. This receptor, this likely switch, exists in other types of reperfusion injury. We know it can drive lung injury. We know it's on the macrophage. So let's look at this receptor. The problem is there are a number of strategies that you can use to target this, and all we wanted to do was selectively and transiently knock down this receptor in one cell in one organ in the body. That's very, very hard to do. The other problem is the lung transplant is the only organ we transplant that remains in constant contact with the environment. Our kidneys are not exposed, our livers, our hearts are not exposed, but the lung is. So the lung needs to remain immunocompetent to fight off infection. If we block the immune response within the lung, it probably won't get very excited, but it'll get badly infected shortly thereafter because it can't defend itself. So we needed a strategy that allows us to knock down a target in one cell, but only do it for a little while until they got through the reperfusion period and then let it reconstitute so they could fight off infection. So the only way we could do this was by using something called a small interference RNA. It's a gene particle that you infuse, and we showed that it tracked to the circled cells, which are the macrophages only. This took years to develop this strategy, and it was the first time that this strategy had been used in an intact animal. It had just been used in cell culture dishes before that, but we showed it could work in an animal system and target the macrophage exclusively. And lo and behold, when you block toll-like receptor 4 and you neutralize it for a little while, you take away lung injury. You basically take it away. You prevent it almost completely. We used an animal model that modeled the events surrounding lung transplantation and showed that we shut injury off with this strategy. But this is not a clinically meaningful strategy. It just is sort of a teaser because it says now we know the mechanism, but how do we take advantage of that? Well, I was talking to a colleague in pulmonary medicine years ago now, and he said, well, you need to make this more clinically relevant. And remember, this is one of my basic research principles. It needs to be clinically relevant. So he said, that receptor you're talking about fires when it's exposed to certain bacterial products. And in reality, most lung donors are on the ventilator in the ICU with bacteria in their airways. And we have long thought that this means that these organs will be dysfunctional when you combine them with a reperfusion period. In other words, that the injury would be worse. So he said, give them a little bit of this bacterial product, and the one that I'm going to talk about is called LPS. And that's going to make injury worse, and you'll be that much closer to at least understanding the mechanism that underlies this problem. And this is what I'm talking about. 80% of lung donors that we use have bacteria in their airways. We do this knowingly, and we did it for years with a little bit of trepidation, thinking that maybe these were lungs that should be discarded. We're very aggressive here, and our results are excellent, but around the country, many more conservative programs were throwing these organs out. They were going to waste. So what we postulate is that with pre-stimulation with that LPS, we'd activate the receptor and a subsequent signaling cascade that would result in a pro-inflammatory release of proteins or cytokines, recruitment of inflammatory cells, and ultimately tissue injury. We thought that the additive effect of the bacterial product exposure and the oxidative stress would worsen lung injury. That was our theory. But what we found was quite the opposite. What I'm showing you here is a graph basically of injury severity when animals were pre-treated with a sliding scale dose range of this LPS, this bacterial product. And at the bottom, you can see that at a reasonable dose, this is the amount of bacterial product that you see in a typical donor, injury went away. Injury didn't get worse, injury went away. And when we looked at inflammatory cells that were migrating into either the tissue or the airway space, they weren't there anymore. So this is a fortuitous finding. This is what we're talking about when you say embrace unanticipated findings. This bacterial product was not only not injurious, it was highly protective. We're able to deduce ultimately because when you do something like this, I mean, you could assume that putting something that would be presumably dangerous into a clinical construct would be met with some hesitation. So you have to prove how it's working. So we proved how it's working by spending the next number of years uncovering the signal pathway that's actually modulated by pre-exposure to this bacterial product. And we showed that the pro-inflammatory signaling cascade is knocked down and the protective cascade is spooled up. We ultimately found that if you target certain proteins within the protective pathway, that the lung protection also went away. So we figured out that it worked, which was a surprise in the first place, and then we figured out how it worked. So we now had precedent to take this forward. Well, is this clinically relevant? Of course it is, because this is highly protective against reperfusion injury, and this is rather inexpensive to produce bacterial products like this. We are on the verge of taking this to the next step, and the step after that is a clinical trial that could change the way we treat all lung donors. All lung donors. We also were able to show definitively that not only is this presence of this bacteria not a liability, it could mean that these are preferential lungs. Maybe these are the lungs that have some advantage physiologically, and we want to definitely avoid discarding them and potentially use more of them. And most importantly, as I said, the lung is a vigorous inflammatory responder, but these pathways are active in all organs. Sure, we mapped it in the lung, but these same pathways can be mirrored in the liver, in the heart, in the kidney, in the small bowel, and all these problems with acute transplant dysfunction could potentially be mitigated by something as simple as treatment with a partial product that's derived from a bacteria. Could cost pennies to make and could save lives across the world. The problem is that our research grant, which takes this to the next level, expires in about 6 months. And the funding climate at the federal level is poor. So we know now that we're very vulnerable, and we're looking at alternative mechanisms of funding in order to take this idea across the finish line so we can actually start to help patients. And through some generous support of some people on the transplant commission here, I've actually established a Twitter page and a Facebook page, and you'll be able to follow the progress of the laboratory and the transplant program in general. Thank you very much for your attention.
What will Dr. Mulligan do to fund his research? I think that's a perfect segue into crowdfunding. Now, many of you have probably heard of crowdfunding. Believe it or not, it's not a new concept. Some of you have probably heard of Jerry Lewis and some telethons that he did. Or perhaps during the holidays, you hear a little bell jingling and you see the Salvation Army bucket. Both ideas of crowdfunding, where each person contributes what they can individually to pursue a greater cause. And that's exactly what this crowdfunding is about. So when you think of technology crowdfunding, Kickstarter is typically the website that comes to mind. And actually, it launched in 2009, and as of last week, just passed the $1 billion pledged mark. So it's become a very powerful tool to fundraise. And that, coupled with the fact, as Dr. Mulligan alluded to, funding for research is getting more and more difficult. 15 years ago, approximately 30% of NIH grants were funded, and now that's down by at least half, and in some health categories, even lower than that. So really, the question becomes, how can we leverage this existing idea of crowdfunding in the medical research realm? So how did Consano, a 501c3 crowdfunding platform, manage to attract a world-renowned thoracic surgeon as one of our first projects? Well, clearly because he's an innovative thinker. So we first heard about Dr. Mulligan's work through one of our scientific advisory board members who happened to study under Dr. Mulligan here at University of Washington. So once we heard about what he was doing, we immediately contacted him and asked, would this be something you're interested in, given climate of funding and, this kind of new connection that you can get with people interested in learning more about what your research is, and he was immediately on board, for which we are very, very grateful. After the initial launch of his project, people started talking, what is this? What are they doing? He garnered some great press from King 5 up here in Seattle, talking both about his innovative research and getting that to be part of the conversation, as well as the innovative way he was using to fund the research. So to date, as you can see from Dr. Mulligan's project page, he's raised $12,800 from 57 donors. So 57 donors. I'm kind of looking around here. I was told that there are approximately 300 or 350 people here tonight, maybe a few less given the beautiful weather outside. And, I would just like to pose the question, what if each of you donated what you could to support Dr. Mulligan's project, and not only that, shared it with your friends, your family. Now that he is linked up on social media, you can tweet, you can Facebook, all these new tools to spread the word. Think of the power behind those types of numbers. And as we speak, I'm going to refresh this page if I can figure out how to do that and see what's happening. So we're at 12,800 now. Let's just take a peek here as it refreshes. 12,830, look at that. That's right, you guys, that's what I'm talking about. Very cool. So, how did this all come about? Clearly you saw the picture of me with my haircut gone wild, as the preschoolers in my daughter's class called it. Clara's mom, why did you choose a bald haircut? I did not choose that. Unfortunately, in 2011, I was diagnosed with breast cancer. I was 32. I had a 3-year-old and a 5-year-old at home at the time, and clearly, my world was turned upside down. So I went through my surgeries and treatments and really came out hoping to support medical research that might help my 2 daughters avoid walking this same path of breast cancer now that they're at a higher risk. And as I started to kind of explore ways to do this and to get involved, really I wasn't finding the kind of transparency that I was looking for. I wasn't finding an easy way to direct my donation to projects that might specifically impact my daughters, whether that was genetics or vaccines. And most importantly, I wasn't finding an easy way to remain connected with the research I was supporting. So Consano has aimed to address all three of those things and provides transparency on several levels. First and foremost, the projects are written in language that us non-scientists can understand, which already kind of gives us a window into what's going on without, 8 of the 10 words in the sentence being very, very long and very difficult to pronounce. Secondly, you can literally direct your money to whatever specific research matters to you. So we're shifting the paradigm from you give, they decide, to you give, you decide. And I think that's what I was looking for when I was looking at my donation options. So most importantly, I was looking for an ongoing connection to this research I would be supporting. And again, Consano has created an easy way to maintain that visibility to the impact of your donation through quarterly updates from the researcher on their research page. And you can see there's an update tab, overview researcher updates. A very quick and easy way, both for the researcher who, frankly, we want to be doing the research. We don't want them spending a lot of time having to be outside the lab. As patients, we want them to be doing the science. So I think that Consano is really an innovative way for people who are touched by Dr. Mulligan's work to connect and support what he's doing. And I think that's probably best illustrated by reading to you some of the stories people have shared on our website about why they supported Dr. Mulligan's project. And I'm going to try not to get teary. These tend to get to me a little bit, so bear with me. Oli says, many people like me with cystic fibrosis and other respiratory diseases die on the transplant list every year because there are not enough lungs for everyone who needs a transplant. It's important to encourage more people to register as donors, and as this project is doing, find ways to rescue lungs that would otherwise be unsuitable for transplant. It will give people a second chance at life. And Deborah says, I am a lucky and grateful kidney transplant recipient of 8 years now. Without the gift of well-trained doctors, dedicated researchers, and of course, a generous organ donor, I would not be here to continue to love my husband and family, see my daughter married, watch the deer and moose wander in the meadow. With so many people waiting for organ transplants, I want to help with research that will improve the viability of available organs. As part of the transplant community, I can tell you that you never take lightly the renewal of your life. And lastly, I'm so pleased to have here tonight one of Dr. Mulligan's patients, Gretchen. If you wouldn't mind standing up, Gretchen. Gretchen had her lung transplant 18 months ago. And look at her, she looks fabulous. When I asked Gretchen about her thoughts on Dr. Mulligan and the work he does, she said, I appreciated his caring, his attentiveness. I'm grateful because I'm here. I'm breathing. I'm walking. And her husband, Walt, chimed in, I'm grateful to have my wife back. So if these stories don't illustrate the power and the passion that fuels the crowd, I don't know what does. We're so grateful for the amazing work Dr. Mulligan's doing and his willingness to try out crowdfunding with us. Not only does Consano bring transparency to the donation process, but we're building a community of people touched by health issues. Hearing these stories reminds us that we're not alone, that together we're stronger. Thank you, guys.